Namaste students. In this video we are going to learn about the dicot and monocot leaf. In previous classes we learned about the structure of dicot and monocot, stem and root. So now we are going to see about leaf. So on observing the leaf itself, it is we can able to distinguish whether it is dicot or monocot. See here, this is a dicot root. So in this root, in this leaf you can see the venations. They are not parallel to each other. So this is a dicot leaf, whereas in monocot leaf the veins are parallel to each other. So you can see the parallel venation of these leaves. So by this itself we can able to distinguish the monocot and dicot leaf. So now we shall go to the inner structures. See here the dicot and monocot leaf diagram. So from this diagram itself we can notice the difference between them and we can read one by one in detail. So on seeing itself, as I said, in monocot it is parallel venation. So the leaves are in parallel structure, whereas in dicot the leaf is not parallel. Now we go to the epidermis. So epidermis is the outermost layer. So the leaf has the upper epidermis and lower epidermis. So in both monocot and dicot, upper epidermis and lower epidermis are seen. So now we shall go to the upper epidermis of a dicot. So see here, upper epidermis. So upper epidermis is the outermost layer and it is made up of single layer of parenchyma cells without any intercellular space. So these outer wall cells are cuticularized and stroma is less in number. So as I said before itself, cuticle is present in the shoot system, and here also it is cuticularized and stromata are less in numbers. Next one is the lower epidermis. So lower epidermis is the lower side of the leaf. So it is also made up of a single layer of parenchymatous cells with thin cuticle and it contains numerous stomata. So when compared to the numbers of stomata, it is lesser in upper epidermis and more in the lower epidermis. So here the function is the exchange of gases and loss of water vapor. So it monitor the transpiration level. Now the epidermis of the monocot leaf. So the monocot leaf also has the upper epidermis and lower epidermis, and it is made up of parenchymatous cells. Cuticle is present on the outer wall. Stomata are present on the both the side. Small cells in the upper epidermis are large and they are called villiform cells. So in monocot, some of the cells are bulky and large. So that cells are called villiform cells. So see the diagram. So upper epidermis and lower epidermis in both the monocot and dicot. The next one is mesophyll. So the mesophyll is the middle region between the upper and the lower epidermis. In dicot leaf, the mesophyll is differentiated into two types. They are palisade and spongy. Whereas in monocot, there is no such differentiation. So now we shall see one by one. So first one is Palisade Parenchyma. So these mesophyll is made up of parenchymatous cells. So and the first one is Palisade Parenchyma. It is found below the upper epidermis. So Palisade Parenchyma is seen below the upper epidermis, whereas Spongy Parenchyma is seen at the region of lower epidermis. So the cells are elongated and they have number of chloroplasts. So in Palisade Parenchyma, they have large number of chloroplasts. The cells do not have intercellular space. They takes place in photosynthesis. So this Palisade Parenchyma are arranged closely to each other and they takes place in the photosynthesis. The next one is Spongy Parenchyma. So it is found below the Palisade Parenchyma. The cells are almost spherical or oval and they have lot of intercellular space. So when there is a intercellular space, they helps in the gaseous exchange. The next is the monocot leaf. In monocot leaf, the mesophyll is not differentiated. So, the cells are irregularly arranged with intercellular space, and these cells contain chloroplast in it. Now we can go to the vascular bundle. So vascular bundle is nothing but the innermost region. Vascular bundle comprises of xylem and phloem. So vascular bundle of a dicot leaf is in the midrib region, it is larger. So when you see a dicot leaf, you can see the midrib. So in this midrib region, they are larger, and vascular bundle are conjoined, collateral and closed. Each vascular bundle is surrounded by a sheath of parenchymatous cells. So when something is surrounded by a sheath, that is called a bundle sheath. Each vascular bundle consists of xylem towards the upper epidermis and phloem towards the lower epidermis. In monocot leaf, the vascular bundle are large in numbers, some of which are smaller and some are larger. So there is no specific regions, whereas in dicot the midrib region is larger and other region are smaller. So likewise in monocot it is not so. So each vascular bundle is surrounded by parenchymatous bundle sheath. Here the vascular bundle is conjoined, collateral and closed. So xylem is present towards the upper epidermis and phloem towards the lower epidermis. So see the diagram here. So you could see the xylem and phloem. So the pink color circular cells are called the xylem and blue color cells are called the phloem. So phloem is towards the lower and xylem is towards the upper epidermis. This is in the both the cases. So now we shall have
the monocot leaf. So in the previous videos we have learned about the dicot root and monocot root, dicot stem and monocot stem. So we are learning about the dicot leaf and monocot leaf. So first we will see about the dicot leaf. The dicot leaf is also called as dorsiventral leaf. So similarly, we'll be seeing about the various structures present in the dicot leaf. So there are three structures present in dicot leaf. So first one is the epidermis. So epidermis is very similar, very different from the roots and stem of the leaf. So the leaf of epidermis has upper and lower surface. So the upper and lower surfaces of our leaf are covered by epidermis. So the upper epidermis is called as adaxial epidermis, and the lower surface of the epidermis, that is lower epidermis, is called as abaxial epidermis. The percentage of stomata, the amount of stomata is more in abaxial epidermis compared to adaxial. So this is the parenchymatous cells are present. Coming to the second, which is the mesophyll. Mesophyll is the layer between this abaxial and adaxial epidermis. The cells are parenchymatous as they take part in photosynthesis. There are two types of mesophyll cells present, which is the first one is the Palisade Parenchyma, and the second is the Spongy Parenchyma. This is close to adaxial epidermis, and this is the main site of photosynthesis, where the spongy parenchyma is very close to the abaxial epidermis, but both take part in photosynthesis. Coming to the third, that is the vascular bundles. The vascular bundles in leaf is present in midribs and veins, and the vascular bundles are covered with bundle sheath cells. So this is about the dicot leaf. So now we'll see about monocot leaf. Similarly, monocot leaf is comprising of three parts, that is epidermis. The first is the epidermis. So the epidermis is in epidermis all the parts are similar, like adaxial epidermis and abaxial epidermis, but in this monocot leaf the stomata, number of stomata, that is the quantity of stomata, is equal in both the adaxial as well as abaxial epidermis, which was greater than in the abaxial epidermis from the adaxial epidermis in the dicot leaf. Coming to mesophyll, the second one is the mesophyll. The mesophyll is not differentiated here, as it was differentiated into Palisade Parenchyma and Spongy Parenchyma in the dicot leaf. So monocot leaf, this is not differentiated, and the cells are parenchymatous, and they contain chloroplast. Coming to the vascular system, that is the vascular bundles. This is arranged in parallel manner, and the bundles are covered with bundle sheath cells.